about a general theme. So I've subtitled my talk Contributions from Queensland to Global Food and Energy Security. I can remember when I was an undergraduate, I think one of the things that inspired me was learning about the fact that there was research going on locally that actually had global significance. And at the time, the big development was the identification of the C4 pathway of photosynthesis out at Indooroopilly. And I know that to me was inspiring in realising that science wasn't something that happened elsewhere. It was something that could happen here. And so I'm interested to explore from our work what contributions research in this part of the world can make to global food and energy security as a theme today. I'm going to talk largely about work from my own research group, but to set the scene, I'll go through some of the broader issues. We all hear about food security and the importance of feeding the world, but this is a complex issue and has many dimensions to it. What I'm really going to talk about today is how we can utilise some of the novel diversity that we have to support agriculture and food production, and this will not only be the natural biodiversity in Queensland, but also some of the genetic resources that we have in the domesticated material that we work with, and some of the unique contributions that can make at an international level. So we have certainly a lot of crop wild relatives in Queensland, and for a long time we felt that Australia was very poor in plant genetic resources, that we really hadn't any crops to domesticate. But as the science has advanced, we've realised that more and more of the secondary and tertiary gene pool that can be accessed with more modern technology is really found here locally. And while Queensland and Australia might not be a great contributor to the primary gene pool of a lot of crop species, it has an enormous amount to offer if we go a little bit further out to secondary and tertiary gene pools. And in fact, probably represents a very substantial global resource in that sense. I also want to talk about some of the unique genetics that's in some of the domesticated crops in Queensland as well. We're also in an era, I think, when there's been very rapid advances in technology. I'm now guaranteeing people I won't write another book on molecular markers in plants, or won't edit another one, because I'm convinced that it moves too fast, that each of the books I've been putting out are really pretty much outdated by the time they're printed, because it really is continuing to be a very rapidly moving field, the whole area of molecular analysis of plants. The drivers around food security, we all know about population growth, and these are the figures we've had for a few years. It's interesting that the latest revisions of these figures are to advise the population forecasts upwards, and that's really probably because of the global financial crisis. People control their fertility when they're affluent, but when we have more difficult financial times, population growth grows more quickly, and we're starting to see that happen. So the challenge of population growth is actually looking like it's going to be bigger than we've been forecasting. But we all know that that's actually a minor part of the challenge. It's the rise in food consumption per person due to underlying growth in affluence in Asia that drives demand for food. But there's a third dimension to this. There's growing population, more food required per person, but there's a move to eat different food. We've got globalisation of diet, and we're now looking at people eating food that's far more demanding in terms of resources and inputs than the food that they would have eaten in the past. So together, those three factors create the food security challenge. But as Mike Gidley would say if he was here, that it's not really just about food security, it's about nutritional security. We need to give people the food that's required to allow them to lead a healthy life. We need to deal with the population health, not just meet minimum requirements. And that creates the demand, really, for our work. It's also important in terms of what we can contribute locally to realise that much of the growth in the population in the world is in the blue line, is in the tropics. So the people that we're going to need to deal with, where we need the agriculture, where we need the food, is in the tropical world. And the temperate world, where all the traditional science has come from to support agriculture and food production, is not where the action is, it's not the area where the need is. And we're uniquely placed, I believe, here in Queensland to play a really important role in that key area of the tropical world. Now, in terms of application of agriculture and dealing with food security, there is a sort of naive view that somehow we've got a responsibility to grow lots of cheap food and send it around the world to feed people. And we see that in the newspapers. We see it even sometimes by farmers feeling that this obligation in some ways is placed upon them. My argument is that our obligation is to provide technology. And a great example of that is Dave Jordan's project, the Gates-funded project that we're supporting development of sorghums in Africa. So our big responsibility is to provide technology to support food security. It's not the food itself. Australia is a relatively small producer of food and will remain so. Our role is in technology exports. When it comes to actual food exports, I think we need to come back to think about Australia and Asia and our opportunities for high-value exports of food into Asia. So I spoke last week at the Australian Agricultural Economics Conference on this theme, and my theme really is that the business model for Australian farming is to produce very expensive food to sell to rich, largely Asian consumers. And then we need to stop trying to undercut the imports and produce cheaper food for the supermarkets, but rather set the benchmark at the other end, require the domestic market to match the price that can be obtained in export. So we need an export parity rather than import parity focused agricultural business. That's a mindset change for a lot of people that are in the business, but it's all about maximising the value for agriculture domestically. So, but how are we travelling in terms of food security? There's many analyses of this, but this recent analysis compares the demand for these major crops with the actual productivity gains that we're achieving. And you can see that this is the proportion of the productivity gain out to 2050 that's actually been delivered by current industry. And you can see that in maize and soybean, where more than half of what's required is being delivered, we're doing okay. But in crops like rice and wheat, we're really falling a long way behind what the demand curve is suggesting. So, for example, in wheat, and it's probably the worst, we're now growing at probably less than 1% per annum in total productivity, whereas we'd need to be above 2% to meet our projected demand. So what I see here is that this red gap is the innovation gap. We have to provide technologies, research and development to fill that gap and to deliver the demand. I'm sure we'll do it, but it says that that gap now is quite large, and it's quite large for our two biggest commodities, or food commodities. So I'm going to talk now really about species of interest in my own group and how some of that work will contribute to these bigger global challenges of food and energy security. So in relation to food, I'm going to talk about really what we're doing in relation to rice and wheat, those two major commodities that represent such an important part of human calories and human diets. I'm going to talk about a couple of species that we're targeting that really have more economic drivers in terms of agriculture. I work with coffee and with macadamias. And I'm going to talk a little bit about our energy crop work with sugarcane and eucalypts. So I hope in what I say, I'll provide examples of how research here in Queensland can really make a difference in terms of delivering global food and energy security based on these examples. So first of all, I'd like to talk about rice. If you go to Tokyo, and this is a field on the Tokyo plain over here, you'll find that you can become acutely aware of the challenge of feeding a population like the Japanese on rice, because there's no way they could possibly produce enough rice to feed the population domestically. But if you come to North Queensland, here's a scene where we have a waterhole with wild rice growing around it. At this waterhole, I think we probably have the two species indicated in the ensets, and these represent what we now think are two new species that represent new opportunities for global rice improvement. So wild rice in Australia has two big opportunities for it. One is that it could represent a novel crop, and there's growing interest in producing Queensland rice as a crop, and that certainly has a limited value. But the big opportunity is to use the genetics of this material to improve rice globally. The species depicted is Oryza australiensis, quite a large plant that survives in the dry season by dying down to a tuber. So we have very diverse material in rice, but much of it, particularly the bottom lines here, are represented by species that are found only here in Australia and are particularly diverse in northern Queensland. So this is what we thought was the distribution of rice a few years ago. These are the herbarium records for the 4 species we then recognised as being present in Australia. And you can see that rice comes down to Townsville or about there in Queensland as a native species. But as we've looked a bit more closely, we've identified that really there is more complexity in the populations that are out there. And we've been particularly interested in understanding the relationship between Asian and Australian wild rice. When I first started talking to our colleagues in Asia about this, the big question they always asked was, how did the rice get to Australia? How did it get from Asia to Australia? And the analysis we're now doing suggests more and more that the question needs to be reversed. Really, the question is probably, how did it get to Asia from Queensland? Because the evolutionary history may be that it was here first. Certainly the diversity is consistent with that being a possibility, and we've still got more work to do to establish that, but it's just as likely that it started in Australia. So, as I mentioned, the Oryza australiensis forms this tuber. There's lots of adaptation and traits here that could allow us to develop rices with much greater tolerance of abiotic stresses. Our initial examination of the genomes of these species showed us very early that the Australian populations were quite distinct from those we found in Asia. The green is the Asian group, including the domesticated species. But we had a real challenge because the Oryza rufipogon in Australia, which is meant to be the progenitor of cultivated rice, and Oryza rufipogon in Asia really are part of quite different clades in this phylogenetic analysis. So we realised that the classification was wrong, and we're in the middle now of really trying to revise our analysis there. We have in this an annual species, a couple of perennials from Australia, and an Asian species. The interesting thing is the Australian material is bigger, has a whole lot of attributes that will make it quite attractive for domestication and food use. Marta, who should be here, has just recently sequenced the chloroplast genome on one of the key species, and this establishes quite clearly that this is a very distinct taxa that may be, in fact, the ancestral species to the whole clade that was domesticated subsequently in China. So we now have a model where we're adding extra species in the Australian group, and I expect that that will continue. That suddenly Australia is an important source of diversity for rice, maybe the most important source of diversity, because in Asia there's not much wild rice left. Where rice was growing before, it's been displaced by cultivation of rice, in many cases for thousands of years now. Where there are wild populations, they've interbred with domesticated rice, and so when we look at them, they're actually domesticated. Genuine wild rice may only be found easily in the Australian populations, and so we've got a new reservoir of genetic diversity. We're just at the beginning of capturing for rice improvement. This is what we're currently calling species A and species B. We've got Brian Simon from the herbarium working. I just had an email from him this morning working on trying to describe these as potential new species. We have, just as we're discovering these species, a renewed interest in growing rice in northern Australia, so we have to move quickly to capture and conserve these populations before they're actually displaced by the same fate that met rice in Asia, that is the fate of being displaced by domestication. And here we have a great example where the cattle get in and eat the rice in what's probably the type location for one of these species. There's not much left after they've eaten it in the dry season, and there's a sugarcane field that's displaced one of the other major populations. So we have challenges of describing these species and putting in place appropriate conservation strategies. But we have very large populations of rice in northern Australia, to the extent that I believe this may explain why agriculture was slow or didn't develop, in that Indigenous populations would have been able to access fields of rice without having to plant it, and creating in Australia uniquely a lack of incentive to develop agriculture. So we're beginning to again understand and try and look back at traditional uses of rice in Queensland. This has led us to look further afield, and Peterson, who's here, has come to join us to look at what's happening in Africa, the African wild rices, and trying to understand the evolutionary relationships between rice in Africa and rice in Australia. And I think by putting it all together, we'll eventually have the big picture of where to look for diversity and how to manage capturing of genetic diversity in rice improvement. But I think it's really, this move has been catalysed by what we've discovered in northern Australia and what the questions that it's asked about rice genetic resources globally. What we've found by looking at the whole genome of these, and now of course we can do that, is that there are, in the rice genome, in domesticated rice, and this is an analysis where we've looked at the 12 chromosomes of a number of domesticated rice samples, we can find what we call genome deserts. The red areas are areas where we don't find diversity in the domesticated gene pool. And you can see this very large area, about 5 million base pairs on chromosome 5 there, where there is just no diversity in domesticated rice. Another area, about a million base pairs up on chromosome 1. So what we're about being able to do now is deliberately bring back diversity into those areas where we've lost it in the process of domestication. And we can be very deliberate because we can now work at the whole genome level in analysing that. So the Australian rice's contribution in this area, we can see if you look at the top panel, that flat area in the brown is domesticated rice, where we see no variation across those 5 million base pairs. But you'll look at the green line is the divergence of Asian wild rice, and you can see a little bit of variation in that size, those areas, but look at the blue. The blue are the Australian rices, and show how much diversity we've got at the nucleotide level along that chromosome. And the fact that we have got a loss, it seems, some loss of diversity in that chromosome region, even in the wild, suggesting that the origin of some of these deserts may not just be domestication, but may be selective sweeps that have happened in evolutionary history. So we're beginning to be able to use the Australian populations to distinguish between domestication-related loss of diversity and losses of diversity that have happened through natural processes of selection. And we're now focused on all of the domestication genes in rice, and this is allowing us to have much more deliberate approach to domesticating new taxa. Another area that we're focused on extensively in rice is understanding the relationship between the genes and the starch composition. Starch is very important in terms of human health, and we've made a number of discoveries of key genetic manipulations, both in the wild, that have happened in the wild, but also under human selection, that influence potentially the health impacts of consuming a carbohydrate diet based on rice starch. And the latest discoveries from Peterson's work in the African rice suggest that the domestication of rice in Africa took a very different path, and it may explain some very novel aspects of these rices in relation to health. We also have new evidence that domestication in Africa involved interaction with Asian gene pools, so for the first time we may have some more involvement of humans in moving rice around in the last 10,000 years. So hopefully in another few months there'll be a very big news story there about what's happened in domestication of rice. Done a lot of work on starch diversity, but one of the examples in rice that I think is most important is our discovery of the fragrance gene. Fragrance doubles the value of rice. Most people know basmati rice or jasmine rice. What we've identified is the mutation. It's a loss of function of an aldehyde dehydrogenase that leads to accumulation of 2-acetyl-1-pyrroline. We got this very quickly once we had the whole genome sequence for rice. But this is a really great example of how human selection has been in conflict with natural selection, because this gene is in a pathway that's upregulated in plants in response to stress. So that proline metabolism, many of you will know, is upregulated in plants when they face stress. It's disrupted in these plants, and it explains for the first time why fragrant rice can't be grown very successfully in many of our environments in Australia, because it's actually susceptible to abiotic stress, particularly to salt stress, but also to drought stress. And so this now gives us new clues as to genetic strategies for producing high-value, high-yielding rice for the first time. We can double the value of the rice, but we can also think about genetic strategies to bring the yield back to a level that will be acceptable in some of our environments. So being able to work at the whole genome level allows us to look at the genes that are associated with each of the traits that humans have selected for, and working out new strategies to work with them. So we've got great tools, and we're moving towards the ultimate solution where we'll be able to select for all of rice quality traits, for example, in a single DNA analysis by measuring all of the alleles that we need to get all of the characteristics. Rice quality is complex, but probably not so complex that we won't be able to get an almost complete molecular solution. Part of this is leading us to look at domesticating new crops, and I think Queensland has a number of species that we could work with. One that we have worked with is Microlina. You can see the dark coloured grain there compared to domesticated rice. We're selecting now for bigger grains and for other attributes, and there's been a company spun off to Alpine Rice to commercialise this as a rice that can be grown in dry areas and that will be consumed in the way that we normally consume rice. So rice is a big opportunity. Wheat is another very interesting one. Wheat is such a wide range of end uses. Our focus is particularly on things like bread and noodles, which is what's important for Australian wheat. The big area of research that we've had is to look at trying to explain through analysis of the transcriptome those genes that are important in end-use quality of wheat. Wheat production, as I've shown, is not keeping pace with demand. And we have to ask the question why. When I talk to wheat breeders and ask them about their high-yielding wheats, they all have high-yielding wheats, but they don't grow them commercially because they can't be used to make bread, or they have defects in quality. So it's clear that we could very quickly increase wheat yields globally by at least 50% if we could remove the constraints of the end-use quality requirement that really imposes a major restriction on genetic gain in wheat. So our focus has been on working out how we can manipulate the genetics of wheat to achieve end-use quality, hence facilitating the rapid adoption of the already existing and adapted high-yielding varieties of wheat that are found in most wheat breeding programs internationally. And Queensland wheats have proven to be very interesting. We've looked at the transcriptome of about 90 wheat varieties now in some detail, and we're trying to relate that to things like bread quality. And we have found that in a series of Queensland wheat varieties, we have a number of novel alleles for genes that hadn't been discovered before, that we find at the transcriptome level, that seem to explain the bread-making quality. So we've had a great run of prime hard wheats in Queensland, going back quite a number of years, and we can now follow in that lineage the inheritance of the expression of small proteins that seem to be critical for bread quality. So we now have an answer as to why many of our wheats in Queensland have been so successful and have been grown so widely across Australia and have been used internationally. Really, this was all achieved by blind selection for bread quality, but now when we look behind it, we can see that there is a potential genetic explanation. This particular allele is not common internationally, and so it represents something that was picked up in breeding selection here in Queensland and hasn't been adopted more widely. So we're yet to do all the definitive experiments, but adopting selection for this gene or this particular allele may be quite valuable globally in improving wheat productivity. So we have a focus on bread quality here, but we're also working in India on chapati quality and trying to understand how we breed wheats that might actually make both of these products. At the moment, they have quite different requirements, but in being able to move germplasm between India and Australia, we need to be able to understand how to adjust from a bread-making wheat to a chapati-making wheat in either direction. Now, moving away from the food crops, I wanted to say a little bit about some high-value crops, and coffee is one of those. Coffee is one of those very high-value traded commodities. A lot of it's produced in developing countries, a lot of it's consumed in developed countries, so it's a way in which a lot of money moves from rich countries to poor countries. It's economically really quite important, and it is a product of quite high value. We've been interested in coffee for some time, and from an Australian point of view, we're now working with companies that want to produce very high-value coffee. We have a project where the goal is to produce the world's most expensive coffee from Australia. That's a great project goal, to make the coffee more expensive than anyone else's. Clearly, we have to come up with some characteristics that make that coffee really good, but we're starting from a good perception. People already have a reasonably good view of Australian coffee, and if we can work with that and have people that are prepared to be outrageous enough to put very high prices on it, we can try and come up with something that can justify that. But to do that, we're trying to understand the coffee genome in detail, and with Heather Smythe is here, we're exploring the genome in terms of control of all the attributes that influence quality from the consumer's perception. We have been working with the Coffee Consortium, and we've just got the paper submitted to Science this week. The most interesting thing about the coffee genome really is probably the caffeine. The caffeine story is very interesting. So caffeine production, it appears, has evolved separately in tea and in coffee and in chocolate. And that's quite interesting, that they've evolved from different gene family members. So this is an independent evolution of caffeine production. The great mystery still is, why caffeine? I don't think we have an adequate explanation of the biological role of caffeine in coffee. We know a lot about its effect on humans and animals, but why is the plant producing it? It doesn't seem that we have a clear answer, but whatever's causing it is a very strong evolutionary imperative, because it's evolved independently in different families of plants or different groups of plants. And humans have been very good at using those and adapting them for use. I think one of the suggestions is that bees might need to get a caffeine fix in the morning, and so repeat visit to flowers that give them caffeine. But that's just one of the many theories that are about it as to why plants might have caffeine. But what we've got, in addition to being bold enough to try and produce very expensive high-quality coffee, is we do actually have some genetic resources. We've got a new species from Cape York that we're just sequencing. This is a zero caffeine genotype. Some people will say that's fairly useless, but it's actually really important to understanding the genetics of coffee, because we're now going to be able to tune any particular caffeine content we want, because we will have really now, I think, a complete understanding of the genetic control of caffeine in the plant. That work with the wild material brings me to talk about comparative genomics, and we recently compared the genomes in a collaboration with the University of Copenhagen and Washington State University. We compared the peach, the cherry and the almond genome at the whole genome level. And what that's allowed us to do is to understand the genetic basis of a whole lot of traits in these species. So these species are close enough that comparison of the species can tell us about some of the key genetic differences. We understand a lot more about what controls ripening in the peach, because it's got a whole lot of genes that are missing in the cherry, because the cherry doesn't undergo the same ripening process. We can understand now the genetic basis of bitterness in the almond, because we can compare the genes across these species. So comparative genomics is very powerful, and we're wanting to use that in a number of systems, including the macadamia. Macadamia, I think, is not really the Queensland nut, it's the Brisbane nut, because if you look at its distribution, it's around Brisbane, north and south. We really are in the centre of the distribution here. We've got a first draft genome sequence that we've put together, and we're now looking at resequencing all of the wild relatives to understand that diversity and what we can do with that. And we're now in negotiation with the Chinese Academy of Sciences about a new project with them to really look at understanding some of the genomics of some of the attributes, not only in macadamia, but in a whole range of fruit and nut species that might be useful in developing new high-value products. Macadamia, we've got a big effort to improve its productivity, but we not only have to improve its productivity, we actually have to improve the quality of the product. We see macadamia being planted in Brazil, in South America, in South Africa, in China, through southern China. So there's a lot of competition coming for the local industry. We brought the macadamia back home from Hawaii, where it was domesticated, and so we're working with the Chinese who are trying to bring the kiwi fruit back to China in the same way and develop superior products that we can produce in this market that continue to give us some sort of advantage over a world that will be increasingly producing the standard product, if you like. Now, turning from food, I just wanted to say a little bit about bioenergy. And I've had a long interest in this area, and the drivers I see for bioenergy are economic. Oil's expensive. If we can replace it, there's a real economic advantage. Climate change, avoiding carbon emissions is important. That's more or less important depending on the politics of the situation. But security of energy supply is also a very important issue, and it's an key issue for Australia. The economic argument is strong. Australia's run out of domestic oil. The cost of importing oil is in the tens of billions. Every bit of replacement we can produce from domestic sources is clearly an economic opportunity. But there's also a security imperative, because our oil now comes in ships from overseas, and we have very limited short-term supply. If war broke out, we would have cars that we could use for the next week or so, but maybe not for the next 2 weeks. So we really are critically dependent on a continuous supply of oil from the Middle East. To the extent to which we can produce some alternatives locally gives us that energy independence. And there's been a strong interest in the Pentagon in partnering with Australia in this area because of the military links and the interests of the US in being able to operate, particularly in the Western Pacific. So we're looking to develop technology to convert all of the carbon in the plant, the biomass, through to a range of high-value fuels. There's great interest, of course, in the aviation fuel because there's no real alternative. We can't really use an extension cord to get across the Pacific. We have to have a high-energy fuel. Batteries really aren't going to work in planes. They may in cars. We've got a big local interest also in a supply of diesel. We've now formed a consortium supported heavily by the Joint Bioenergy Institute, which is the Department of Energy funded activity that's really allowing us to develop new feedstocks, particularly for bioenergy production in the tropical world, particularly in Queensland. Clearly the US Navy would like to be able to source from Queensland fuel for its Pacific fleet. It's a great commercial opportunity for Queensland agriculture if we can do that. So our future biofuels project has established plantings at Gatton. We have demonstration plantings of a wide range of energy crops, and that range is growing all the time. We're developing new techniques for understanding the composition of that biomass and how we might manipulate it to make more efficient the conversion of that biomass to fuel. And that's the main focus of the research, how to modify the composition of the biomass. But we're doing that by really working at the whole genome level again. We're using genomics linked to advanced chemistry to try and understand those links between genetics and chemistry. Sugarcane is clearly an obvious major focus for us. We have a significant involvement in efforts to sequence the sugarcane genome, and this will produce a platform that will be used more widely than just in bioenergy. The sugarcane is very difficult to work with, being a complex polyploid system. So it's been great to involve the Department of Energy. They're now contributing significantly to helping us to cover the cost of sequencing this large genome. And with their support, we hope to have some sort of a workable draft by the end of this year. A lot of work going on at a lot of levels there. We've also done a lot of work in looking at native grasses, trying to find something that beats the US switchgrass. But the eucalypts is the other group that I wanted to talk about. The eucalypts do represent a relatively unique Australian opportunity, and the eucalypts is probably the thing that's got greatest interest in the US. Everywhere you go in the world, people realise that eucalypts grow in marginal environments, in areas where they maybe won't compete very much with food. You go to South America, you go to Africa, you go to California, you find lots of eucalypts. We don't have to sell the story that eucalypts are very fast growing. In fact, amongst the most fast growing biomass crops that you could find on the planet, particularly when you consider the sorts of environments in which we can grow them. We have great diversity, bordering on 1,000 taxa, and we're now in the process, because they've got quite small genomes, of sequencing many of these to understand which of these species represent the best opportunity as a source of replacement of oil as a major global woody biomass. We've discovered quite a number of genes already that influence key wood quality traits in the eucalypts by doing association genetics, but we're now working up to the key experiments that will allow us to dissect the control of the key traits that determine biofuel use. And we do know that there is large genetic variation, so we expect in the very first rounds of selection that we'll be able to achieve very large improvements in eucalypts as a source of bioenergy. The chemistry of eucalypts is quite challenging, and a lot of what our effort is directed to, apart from the genomics, is really understanding the chemistry, trying to understand what parts of the chemistry we want to change. It's about knowing which covalent links we need to break to pull the biomass apart. So by understanding the biosynthesis of the biomass, we can understand how to deconstruct it. We work with the deconstruction division in JBay, working out how to deconstruct eucalypts. It's all about knowing how they were put together in the first place. If we can put them together differently, we can make them more susceptible to an efficient deconstruction process. We are using quite advanced chemistry here, and to that we are gaining new insights into what's required. I also want to say a little bit about our work on understanding climate adaptation derived from our work at the genomic level. We all understand the need to make agriculture more able to tolerate variations in climate and maybe adapt to a very different future climate than the one we have now. And I've been involved in a series of studies over the last few years where we've been trying to use genomics to understand how we do that better. And the key way that we've been pursuing is trying to understand how it happens in nature. How does natural selection impact upon wild plant populations? And how does that tell us about how plants would naturally evolve to cope with a different climate? So here's an example where we're looking at wild barley across a valley where we have a wet slope and a dry slope. That's because of the aspect. One population, by looking at the genome, we can say which genes are different in the dry area compared to the wet area. There's free exchange of genes here, so we should be looking at selection across those microenvironments. We've been doing this also in a vertical sense, so we've been looking at current populations and then going back to seed that was collected 30 years ago and analysing that seed and saying what genes have changed in that population where it's got 1 degree warmer over the last 30 years. How has that changed? And we've also been trying to bring that closer to home. We have a distant relative of rice, so we can use rice genomics to help us, where we looked from Melbourne up over the mountains and back down to sea level, and asked the question, what's changing in the genome in those different environments? We get more or less the same answer here that we got with the wild barley, that in the more stressed environments, we see greater diversity. This is a little bit counterintuitive, but what we see in the very favourable environments is the dominance of particular alleles in the population, whereas in the more marginal environments, we see greater diversity, maybe greater heterogeneity within those environments, but adaptation to hotter and drier environments is largely associated with a greater diversity in those populations. But also, quite remarkably, we see that the variation is greatest in the biotic stress genes. So what this is telling us something that we probably already know, but it's really good to see it coming out of the data, and that is that adapting to a higher temperature is not about the temperature per se. Heat stress genes the best allele is probably the same in both of those environments. What's different is the biotic environment. There's different pests and diseases when you go to a higher temperature, and the plant has to adapt to those. So a big part of adapting a crop to a different temperature is not the temperature itself, but it's all the other things that come along with it, particularly the different disease pressures. So we can see great diversity in the biotic stress genes between these populations, and relatively little difference in the allele frequencies in the classical biotic stress genes. And that I think reinforces that argument. We have to keep in mind the whole dimension of adaptation to climate. It's not just about the temperature or the rainfall, it's all the other things in that environment that come along with it. And so we're really trying to coordinate this through a number of efforts to try and integrate what's happening in genomics across species and across traits, because there's lots of effort on individual traits that are being coordinated, lots of efforts on individual species. We're trying to get international collaboration that will incorporate all of that together. And in early January in San Diego, we had a meeting of the SeedSeek Consortium, and the strategy there is to attempt to raise the support and develop the systems to sequence all of the seed collections of all of the major crops. So this is an effort to get a genome sequence for 7 million accessions. That would include all of the major accessions of all of the major crops as a basic resource to work with. Now, this is fairly expensive and radical suggestion, but it will give us the ultimate tools to be able to manage plant genetic resources at a global level, because we'll be able to exchange the data and having at least an approximate genome sequence of everything that we're working with. In that, I wanted to say a little bit about some of the technologies we're developing in the lab, and there's quite a few people here that will have contributed to some of this. With next generation sequencing, we can sequence the genomes in rice. It's really easy, such a small genome compared to some of the things that we work with only 380 megabases. But we've had a lot of focus on new methods for identifying plants, and our big focus has been on the chloroplast. You'll see here in rice, it's 134,551 base pairs. We know every one of those bases very well, because every time we sequence a sample, we get a complete sequence of the genome of the chloroplast, and we can now extract that and assemble it. So what we've done is put a lot of effort into developing this as a platform technology for plant identification, and this we are looking forward to have application in food authentication, in food safety, in a whole lot of applications where we can very quickly and definitively, with the same standard technique, identify any plant sample or any mixture of plant samples in a single test where we do a next generation sequencing run on some DNA from the sample, and we use some standard tools to assemble a complete chloroplast and use that as the barcode. So barcoding is something that we're continuing to develop. We just have a major review that's just been accepted in biological reviews, reviewing the move from barcoding at the gene level to barcoding at the whole genome level. But this is a move that's really quite important, and one great example where we first managed to get really effective de novo assembly of chloroplast was in understanding the red cedar populations on the east coast of Australia, where we can actually better manage the biodiversity of those forest reserves. And in doing that transition, new sequencing technologies will continue to be important. We've been looking at a number of new platforms, and I think the great and exciting thing today is that the sequencing technology hasn't stopped, that we're continuing to see new opportunities, and we're continuing to look over the horizon at where we might be in 6 or 12 months' time in terms of our ability at very low cost to extract all the genetic information we need from a plant sample. And we've I think, got real opportunities to drive sequencing costs down another order of magnitude or so from where we are now, and that will take us into a very wide range of agricultural applications, ones that we're just still struggling to fully identify. So I'd just like to end by thanking and acknowledging the contribution of many of the people in my team, particularly the Agnello and the students, many of the students, but also some of the collaborators in the work that I've been talking about. We're building, I think, a fairly effective group in having the capability of analysing plant genomes at a number of different levels, and of really automating and streamlining the data analysis that's such an essential part of this sort of business. Thank you.